Welcome everyone. Welcome to this webinar session of the TN2, the Translational Neuroscience Network, coming to you by Amsterdam Neuroscience. And before introducing myself and my co-host, let me first introduce you with the session we are in, the webinar session of Zoom. So the webinar session of Zoom is actually only the speakers and the host are visible for you, and you as an attendee are muted. But of course, you can interact with us, and we have the buttons on the bottom of the screen. Please do not use the chat button; that is for the organization. But you can use the Q&A button on the bottom of the screen, and you can submit your answer, and you can also upvote your questions from other attendees. So please use those to connect with us and the speakers. And of course, we will select the questions you have. And discuss them with both speakers. Another issue of this webinar is the adjustment of the visual presentations. You can adjust it to your own preferences, of course. You can see below in the screen. If you put your cursor right on the boundary of the presentation mode and the screen of the presenter, you can actually modify it according to your own wishes. So hopefully that is all clear to you. So let me give you a warm welcome to this webinar session. I am Mirjam van Zuiden. I'm a clinical pharmacologist at the Department of Anatomy and Neurosciences, and specialized in neuro and psychopharmacology. And of course, I would like to introduce my co-host, Sabine Spijker. She's professor at the Department of Molecular and Cellular Neurobiology. She's a team leader of animal models in psychiatric diseases. And of course, she is here because she's the program leader of the research program of mood, anxiety, psychosis, stress, and sleep. So before we go into the program, something about the TN2 first, the background. So the TN2 stands for Translational Neuroscience Network, and our goal is to form a bridge between fundamental and clinical neurosciences. And we have annual conferences and webinars and of alternating topics, and always are focusing on the field of neurology and or psychiatry or in between. And this year we have webinars brought to you every month, and they are focused around the research programs. So this is the research program edition of the webinars. And so we have 9 webinars of each research program. Sabine Spijker will enlighten you a bit about that. And we have, of course, another webinar coming up, and we will do these webinars until June 2022. So feel free to join, and of course, spread the word, and let's connect in the TN2 network. So I will now give the word to Sabine Spijker.
Thank you, Misha. So welcome.
You're still unmuted, Sabine.
Oh, thank you.
There you are.
Thank you, Misha. So welcome everybody, and of course our speakers also for today. First, a bit on the research program. As you can see here, we have all the pillars of Amsterdam Neuroscience, and at the far right, not the least, you could say there is mood, anxiety, psychosis, stress, and sleep. So what we in fact do is we see that these, all these kind of parts, depression, anxiety, psychosis, they are really important and prevalent mental disorders. What we are interested in is to see, really the biological underpinnings of these type of diseases, shared risk factors. And for that, as you can see also here, we take this approach, like from a lifespan perspective, to take a look at vulnerability to late disease chronicity. We have, like collaborative research projects internally, externally, and I'm together with Christian Vinkers, the program leaders, and we have a set of task force members from different like official institutes like the UvA, Liewe de Haan, UMC, and Mirjam van Zuiden, who is in fact also one of our speakers today. So that is a nice link there. So Then we have the program. So we start with Carmen Sandi. Then we have a few questions and answers after her presentation. And then we move to Mirjam van Zuiden. And I think the really nice thing is, as you can see already from the titles, that it's all about individuals and vulnerability and stress adaptation. So I think these two presentations are nicely aligned in that sense. And we didn't really ask them to, but this is just the way it happened. So after some closing remarks, we can then wrap up. But first, in session 1, we have Carmen Sandi, and Carmen Sandi is director of the Laboratory of Behavioral Genetics from the Brain and Mind Institute, the Ecole Polytechnique Fédérale de Lausanne, or also called the APFL, which is probably easier for most of us in Switzerland. And Carmen Sandi, after her PhD at the Ecole Institute, she did several postdocs, had several research positions all around the world, in CERM, Ecole Institute, Open University, University of Bern. And then she established herself at the EPFL from 2003 onwards. She's very active in various panels and societies, like she's the president or has been the president of the European Brain and Behavior Society, is member of several editorial boards, and all related, you could say, to the topic of her heart, namely stress, memory, psychiatric disease. And from all of this, I think, especially since I saw Ron de Kloet as well as one of the members, it's worth mentioning that she received the Ron de Kloet Award in 2018 from the Stress and Health Society. So there you go. Now, she has a long track record of very nice and impactful papers. See only in Science Advances, she has 2 issues in March with different type of work, one in rats, one in mice. So she works really on the mechanistic dissection of stress in animals. And more recently, she also included human subjects to make the back and forward translation of her findings. And I think today she will take us into the individual stress adaptations and the factors involved. So please, Carmen, go ahead.
Thank you very much. Thanks so much for the introduction and presentation. I'm going to share, I think, the screen and see if it works. Yes, thank you also for perhaps making possible that we are going to discuss this topic that it's really at the core of the work that we are doing in the lab, trying to understand how different individuals respond to stress and also to challenges in general, and why there are differences and how maybe we can tackle them. And the topic, I think it's illustrated in this type of images. Many times in neuroscience, we try to understand general principles of circuits that code for behaviors. And this is right. But the reality is that when we look at the individuals, when we look at the individual spread of points in our data, and also when we look at individual experiences, we know that we have very different vulnerabilities or different responses to life challenges, including stress. So our focus in the lab is to explaining the individual and trying to understand why individuals have different responses and vulnerabilities. So in this session today, I think we're going to touch a little bit PTSD with Mirjam's work, but I also will talk a bit about this vulnerability perhaps to PTSD towards the end of my talk. A lot of the interest in our lab has been to try to understand how stress can affect motivated behaviors, in particular situations like the ones we have here, in which we have challenges, perhaps personal or social challenges that require keep going, require motivation, require effort. And here we are very different in how we respond to stress and to challenges. And this is the way we see it. It's probably a good model to understand also vulnerability to develop depression, because we know very well that depression can be induced by exposure to chronic stress challenges. And here different individuals might have different threshold to be challenged by life experiences. So the scheme, I think, can be explained in this way. when we're exposed to challenges, some individuals are more affected than others in the way their behaviors change. And then a lot of the work that is done in many labs, including ours, goes into understanding what maintains the differences once the stress has been induced and experienced. Why would it be that some individuals are affected in one way or another? And in terms of motivated behavior, that maybe some individuals give up while others continue trying to cope with stressful situations. But the focus that I want to discuss now is the possibility that we identify individuals beforehand that are going to have this individual variation in the responses to stress, to chronic stress, to early life stress. And by identifying them a priori, we can look at what could make the difference. So today I'm going to discuss a lot of the work that we've been doing on trait anxiety. You will see that I will elaborate on this, but also towards the end, another of the key factors that we found in the lab that makes a difference when we are trying to understand individual variability to stress responsiveness is individual differences in the reactivity of the HPA-axis, or even better, regulation of HPA-axis or corticosterone responsiveness when individuals are continuously exposed to stressors. So starting by trait anxiety, the reason why we focused on trait anxiety as a normal personality or temperamental predisposition of individuals as a good factor to identify individual vulnerability comes from work that we did in animals, looking at different spontaneous behaviors and also characterizing those spontaneous behavior, or the components of the spontaneous behaviors, and looking at when we classify individuals according to those differences, which ones would be more vulnerable in terms of alterations in motivated behavior. And we observed that those animals that we had classified as highly anxious, according to the behavior in the elevated plasmase or the open field, were the ones showing higher vulnerability. And also in humans, we found in the literature enough support to also identify these individuals as different. One message that I wanted to share today, because I think it's been quite exciting work that we've been developing in the lab, is that inspired by this animal literature and animal work in which behavior tells us whether or not individuals are more anxious or less anxious by how they explore novel environments. A couple of years ago, we developed in the lab virtual reality scenarios that are kind of translated from animal work to humans. And we tried to see if by putting individuals with virtual reality in similar scenarios, we could capture similar components and also predict anxiety and all other types of vulnerability to stress. So in particular, the two scenarios that we've validated and are working quite well in humans are the equivalent of the open field in the rats. So we have an empty room that people can explore, and also an elevated plasmase, kind of an elevated alley that it's equivalent to the elevated plasmase in animals. And here, traditionally, what I think is the same in animals, what we are doing is to look at the position where the individuals locate in the space. And certainly when we do that, the parameters that are important in animals come up as important in humans, like humans that are more anxious tend to avoid the center of this kind of open field. And also they tend to avoid more the extreme of the elevated plasmase. And this gives quite a good prediction. But in fact, if we look at other components of behavior, by looking in an unbiased way, using sensors of different parts of the body and analyzing gait, cycle, also the trajectories that individuals follow, etc., we could apply machine learning algorithms and predict much better, if you want, the trait anxiety. But then we thought that perhaps when we do self-reporting questionnaires, we are not so good in understanding ourselves, and perhaps sometimes it's difficult to answer questions. So we wanted to ask if by phenotyping people with all these multidimensional data in these scenarios, would it be possible to predict their heart rate variability, which is a parameter that is highly predictive of many different vulnerabilities, both in psychiatric, for the psychiatric disorders, but also for neurodegenerative disorders, also for cardiovascular disorders. Heart rate variability when people are exposed to a highly threatening scenario, which is a dark room, which is very, very scary. And indeed, by doing this approach, we confirmed that when we look at heart rate variability, with time, it keeps going down. So confirming that this is highly threatening, and then putting together many parameters from the phenotyping in the other scenarios, we could have a very good predictive model of those individuals that were going to react with more aberrant high rate variability responses, if you want, when they are exposed to high stress. And here we could see that beyond the position, other parameters related indeed to gait, to a burst head movements, etc., altogether gave us an amazingly good predictive value of which individuals are more vulnerable. So this is one message within the, I think, the general topic of the talk, which I think looking at behavior can tell us a lot, and it's something that we are now approaching also in more unbiased ways in the lab, in animals, and also in humans. So since, as Sabine said, we're also interested in mechanistic understanding of all this, we've been using rodents to try to indeed first identify which ones could be more vulnerable, and then looking for mechanisms. So I'm going to now summarize quite a huge body of work from the lab, in which indeed we characterize rats for their anxiety levels using standard tests as high anxious and low anxious. And we see that these already give us a very good prediction of which individuals are going to win when they are exposed to a social competition, like which normally high anxious individuals have a disadvantage and they become subordinate. Also, this high anxiety gives a disadvantage to struggle, if you want, to cope with adversity in the force-win test. So normally high anxious individuals show more floating responses, more coping, passive coping behaviors there. And when looking at where to look in the brain, we mapped different brain regions in their activation when they were exposed to these motivated challenges, and identified a particularly prominent response in the nucleus accumbens, and in particular in the medium spiny neurons of that contain the D1 receptor, dopamine 1 receptor. And we decided to indeed focus in this brain circuit, because we know very well that this is a reward system, that it's very important indeed for exertion of effort, but also it's affected in depression, etc. So there were many different reasons that convinced us to focus our analysis in the nucleus accumbens, not only, as you are going to see, but particularly at the beginning. And what we wanted to know is if we get a lower engagement of these neurons, medium spiny neurons, would it be that a priori there are differences related to anxiety in these neurons? And I think you can observe this by yourself. When we compare with this type of Golgi labeling, the dendritic morphology of high anxious versus low anxious animals, we see that dendritic complexity is lower in high anxious, and also their connectivity, as confirmed by electrophysiology, is also lower in high anxious than in low anxious animals. And doing transcriptomics of the nucleus accumbens, we found that something that really differentiates gene expression between low and high anxious animals is many of the genes that are related to metabolism, but particularly also mitochondria. So I will try to go quite quickly here and maybe summarize this piece of work so that I can continue with the rest. We looked at mitochondria in order to know if there would be differences. And we found that mitochondrial respiration is affected. Let's say it's less efficient in the high anxious individuals. And this goes together with the differences in the expression of protein complexes for the electrotransport complex chain, and also lower levels of ATP and higher levels of ROS that altogether indicated that mitochondria is less fit in the high anxious animals in the nucleus accumbens. And we did also causal analysis to implicate the function of mitochondria in the outcomes of these motivated behaviors. So in order to go a bit further and identifying which mitochondrial molecules could make the difference, we look first at the electron microscopy to get insights from the morphology. And we observed that the high anxious animals have a larger mitochondria, which is a bit kind of swollen. And they also have lower mitochondria and endoplastic reticulum contacts that made us think that perhaps some of the molecules related to mitochondria dynamics could be affected. So we screened for this type of mitochondria fusion and fission proteins. And in particular, we found that mitofusin 2, which is very important for fusion, but also for the contacts with the endoplastic reticulum, is reduced in expression levels in the high anxious animals in the nucleus accumbens, and then performed studies in which we re-expressed mitofusin 2 in the nucleus accumbens of high anxious animals. And I'm going to just quickly tell you that when we look at the parameters, indeed, we reverted from the high anxious to the overexpressed individuals, the mitochondria morphology characteristics. Also, the dendritic arborization was restored with this type of manipulation, connectivity as well, and the behaviors. The animals that were highly anxious before this manipulation became then low anxious, and then they were not impaired in the motivated behaviors. So we continue trying to understand how differences in mitochondria in specific circuits, output from the nucleus accumbens to other brain regions, and also specific cell types are coding for differences in behavior and also vulnerability to stress. And we have exciting new data that will be prepared for publication soon. But something else I wanted to share here is that because we are looking at anxiety, we wanted to know if the mesolimbic system indeed is functioning differently in highly anxious individuals. And we found that the CRH receptor 1 system in the ventral tegmental area, which is the brain region where the cell nuclei of dopamine are seated and project to the nucleus accumbens, is different in terms of differences in the expression of CRH receptor 1 and differences in the reactivity to CRH. And this is work indeed that we published recently in Science Advances by Salakora et al. And you can have a look in. So I think I have around 5 minutes now for this last part. One aspect which is important for us is to know if this is relevant for humans, and the way we bridge the animal and human work for this metabolic focus is by using MRS, magnetic resonance spectroscopy, in both animals and humans, and then comparing if we have similar relationships between metabolites and behavior. And then in animals, then we can go to more mechanistic understanding. One, I think, very nice convergence between what we find in animals and humans is that high anxious mice, rats, and also following stress, individuals show lower levels of taurine, one of the metabolites in the nucleus accumbens. And this metabolite is important for many different reasons, but to make a long story short, it's also related to the activity of complex 1, which we see that it's inhibited or lower in high anxious individuals. And when we look in humans, again, in the nucleus accumbens, with spectroscopy, we see that the levels of taurine have a very good correlation with trait anxiety, meaning that the lower the taurine, the higher trait anxiety. And this is something we are following up in the lab in terms of nutritional supplementations. This is another example in which that it's now under review and also in bioarchive, in which we observed in humans that the levels of the glutathione, the antioxidant glutathione, were predicting the capacity to exert effort over time in a physically incentivized task. And when we looked in animals, in rats, and classified them for high and low levels of glutathione in the nucleus accumbens, we could see that this was related to the capacity to exert effort in a progressive ratio task. And then with manipulations specifically into the nucleus accumbens, inhibiting glutathione, or with a nutritional supplementation with N-acetylcysteine, which is a precursor for glutathione, we could indeed increase the capacity of these animals to exert effort. And this is one example of how we bridge the findings in the lab with maybe translational approaches. So we've tried different type of interventions, from nicotinamide, urolithin-A, diazepam, medium-chain triglycerides, L-acetylcarnitine, taurine, and acetylcysteine. I don't have time to present it, but I think it gives you the idea of how to bridge from one to the other. And in the next 2 minutes or last 2 minutes, I would like to say a little bit about the other component that I announced at the beginning. Could it be that differences in the regulation of endocrine systems already provide differences in the vulnerability to stress and mental health? And the idea for this project came from observations that we have in the lab, that animals that were exposed to peripubertal stress developed a number of alterations when we analyzed them later in life, including aggressive behaviors, attention, different types of alterations. But in particular, we could predict alterations in the social domain by looking at how the animals responded in terms of the adaptation of the corticosterone response to stress when they were exposed to these peripubertal stressors repeatedly over time, meaning that there was a kind of a relationship between the adaptation of the response between the first exposure to stress and the last one, and the magnitude of alterations or behavioral changes that we observed with time. And then we could see that this was related also to alterations or differences in macrostructural changes in different brain regions. So we decided to create a resource, a genetically selected rat line resource, 3 lines in concrete, in which we had animals that when they are exposed to stress repeatedly, they are going to completely blunt their responsiveness to glucocorticoids. Others would have some adaptation and others would not adapt at all. And sometimes they even sensitize. And this is work that we've been doing over the years in the lab. Now we are in much further generations. And what we could see is that indeed these animals are different naturally. Like the high line, the animals that don't adapt are more aggressive. The low line are more vulnerable to early life stress. But in particular, and this is work by Silvia Monari in the lab, what we wanted to know is if now with this line or this resource that we have of individuals that are going to have a normal blunted response of glucocorticoids when they are exposed to stressors, would this be a vulnerability to develop some of the symptoms of PTSD? And indeed, we looked at fear extinction as one of the alterations in PTSD. And we could see that animals that have this blunted responsiveness of glucocorticoids, indeed, they have impairment in fear extinction. They also have some alterations at the level of structure, and particular hippocampus is smaller, which we also know that it's one of the risk factors or alterations in PTSD. And something else that we know from these individuals is that they have a lower heart rate variability, which is also a risk factor for disease. There is more work in the lab ongoing. And again, this would be something that we would be preparing for publication soon. I don't have time to prepare it. I just wanted to give this general overview of what we think makes particular vulnerabilities to stress. So I will finish here. Thank many of the people from the lab that have contributed to this work and you for your attention.
Okay, thank you.
Thank you, Carmen. Great
Thanks.
talk. Yeah, the audience can, of course, put their questions in the Q&A at the bottom of the screen. So if you have a question for Carmen, please enter it in the Q&A section below, and we can select and ask your question for Carmen.
Yeah, so in the meantime, well, people hopefully are submitting their question. So because especially related to the last bit that you showed, you know, like if you have endure some stress and the way you react to this, I mean, normally you would say like, oh, well, the best response is indeed to have stress and then, you know, go down quickly and to adapt perhaps. But apparently your long line is also not perfect.
Yeah, exactly, because I think when we deviate, it's slight aberrations, and there is always a cost. You mean the low line or the high line?
Yeah, no, the low line, because of course,
Yeah.
in a way you could say, you know, like if you adapt to a certain situation, that might be normally seen as a good response, you know. But apparently for these animals, it's also not completely okay.
Yeah, I think it's very interesting to have this line, because we know so little also about the individuals that have low glucocorticoid responses or stress responses to stress, because normally we've been focusing on those that have exacerbated ones and that get affected in a different way. So of course, we know also from the literature in humans that those that develop PTSD normally have more blunted cortisol responses, perhaps not all the typologies, but there is one that is important. So indeed, it's a different phenotype. We are trying to understand why. There are many reasons why they could be different. I think, yeah, many different reasons.
Yeah.
From the way the brain is developing already, because they are different genetically, and that's important, I think.
Yeah. So in the meantime, Aniko Kurosi had a mail, had a question, and apart from thanking you for the great talk, she was wondering of the timing of the effects for the dietary strategies. How long were the diet supplemented for, and how long lasting are the effects you observed?
Yeah, this is very nice, and thank you, Aniko, because indeed in the lab, like in life, we cannot do all the experiments to answer all these questions, but it depends for each of the nutritional supplementations that we are giving, then we have to take some decisions. And for example, now that we are looking at the medium chain triglycerides, one way that we are tackling this is by doing tests in the animals, behavioral tests at different time points. Even if the nutritional supplementation is, for example, 2 weeks, then we start testing a bit earlier to see from when we see some differences. But we didn't look at a washout and for how long afterwards the effect would be there. But normally, indeed, we've chosen different regimes between 1 week and up to 3 weeks, something like that. We are not going beyond this.
Okay. And I see there's another question from Colin Mironuniuc. Well, anyway, Colin. Also saying, thanks for the talk. There's an impressive literature on the mindset, which shows that human perception of stress modulates their stress physiological response. Do you believe that that this is an important factor in the development of these disorders, or is it something that you have considered in your experimental work? So
Mindsets, do you mean the cognitive components? That's the way you would understand mindset?
Yeah, I think it's human perception over stress. So whether you probably...
Yeah, like so perception, I guess it's indeed how it's...
yeah, whether it's good
or bad. Okay, more generally. Oh, okay. This perhaps because it's already going to, in the way I see things, it's going to define a little bit the individual. If the individual is more cautious, like more anxious or less anxious, then normally indeed they are going to interpret everything. More anxious individuals or people are always thinking, oh, we have to avoid this or whatever. So I think this could go very well with the type of work that we are doing in looking at these differences related to trait anxiety or personalities that are over-worrying. So yeah, I really think it can be very important. But it's not only how they understand stress, it's more general, whether or not individuals see stress in maybe things that others don't see as stressful.
So there's a small question indeed from Kay Kultig. Thank you for the talk. And did you also look at the effect of prenatal stress on the endocrine response of the mice in later life?
Yeah, not prenatal stress. We've done a lot of the work with postnatal, sometimes a little bit with very early postnatal, but most of the work the peripubertal period, which is very interesting because it covers childhood and puberty, and it's a very important time for reprogramming of many systems. So we see that it's very powerful, and of course one cannot do all the time points and all the periods, but peripubertal period is very, very powerful in inducing long-term effects of stress.
Okay.
I also have a small question, maybe. I was intrigued by the initially virtual reality experiments with people. I was wondering whether you selected people that have experience with virtual reality, or did you deliberately choose people that are not? Because I can imagine when you are experienced with it, you know that you are in a virtual reality environment and that nothing will happen to you. Whereas if you are quite unexperienced, you might get caught up in the moment of the virtual reality environment and really experience it as a stressing environment. When you look down, for instance, and you see there's nothing under your feet.
Do you have experience, Misha?
No,
okay.
That's why
no, because
I was wondering.
I think your question was making me think maybe you didn't have experience, because I mean, I don't have a lot of experience, but when I tried, I thought, oh, this is not going to work with me because I'm too rational and I'm not going to respond. And I respond a lot. It's incredible how our brain is hardwired to respond what it's experiencing somehow. It's not cognition there, wherever it happens, it feels really like real. But it's true, we take notes. I mean, we record whether or not people have experience, and then we analyzed whether this was contributing to the effects that we have, and we didn't see an important contribution, not significant. But of course, it can affect, so it's something to keep an eye.
Interesting. Thank you. Thank you for your talk.
Yeah.
Thank you.
We will move on to the next speaker.
Yeah, so the next speaker up is Mirjam. And I think she will have a nice follow-up from the work that Carmen just presented. So Mirjam is assistant professor at the psychiatry department at the location of AMC. And she defended a little more than 10 years ago. So she had her anniversary of her thesis what she did in Utrecht. And there she studied biological factors predicting the development of PTSD, depression, and fatigue. Now then she moved to the AMC for a postdoc. And in 2016, she received the prestigious Veni grant, which resulted in her assistant professorship. She also received a Narsat Young Investigator grant, and since 2020, she's active as a project leader of a long-term grant from Zonneweg GGZ, which is called 2 ASAP. And there she studies prognostic screening instruments to predict those at risk for developing PTSD. So you can see already from what she did before, also what she's doing now, that vulnerability and prediction are major topics of her research. And today she will tell more about neuroendocrine vulnerability factors for trauma-related psychiatric disorders. So with that, I would say, Mirjam, please share your screen and share your results with us.
Yes, Sabine, thank you for the kind introduction. And I'll start by sharing my screen. Something's around. Okay, great. So this is a tough act to follow, but I'm very happy to be invited to present after Carmen here. And I hope we have some nice complementing stories. So today I will focus on findings from studies that we've done in humans only on traumatic stress and post-traumatic stress disorder specifically. And those results are obtained from large observational cohorts of recently trauma-exposed individuals, and also from experimental studies investigating what we call analog trauma paradigms, So I hope there will be a nice complement as to what Carmen just told you about. So I wanted to start actually by giving a definition of trauma, because I know everybody has a feeling of what trauma is, but sometimes definitions people use can differ a little bit from how it's considered really in the field. So traumatic events, they can be considered the most extreme form of stressors that can be occur. Obviously, there's a whole range of stress severity, and we consider traumatic stress to really be at the most extreme end of it. And we consider stressors to be traumatic in case of exposure to actual or threatened death, serious injury, or sexual violence. But the prevalence of trauma is actually quite higher than many people expect. So in 2017, the results were actually published of a global survey on the prevalence of trauma and PTSD, and it was done in more than 68,000 representative adults across 24 countries, including the Netherlands. And it was actually observed that trauma exposure is quite high. So 70.4% of all participants really said that they had at least experienced one traumatic event throughout their lives. And the mean number of trauma exposures was actually 3.2, so different types of traumas. So trauma is quite prevalent, and that's a problem because a history of traumatic events is actually the most consistently found risk factors for all types of psychiatric disorders. And acute traumatic events are also a direct trigger or tipping point for the onset of new psychiatric problems for a substantial subset of trauma-exposed individuals. And this includes anxiety, as was just discussed, but also depression, psychosis, substance abuse, and also mind main focus, PTSD, post-traumatic stress disorder. And PTSD is interesting because it's actually the only potentially chronic psychiatric disorder for which exposure through a traumatic event is really a diagnostic criterion. So it may be a risk factor for other psychiatric disorders, but it's really a prerequisite to PTSD diagnosis. And also one of its hallmark symptoms that really distinguishes it from other disorders is that there's common involuntary intrusive re-experiencing of the memories of the traumatic event. So the symptoms of the disorder are very directly linked to actually the content of the traumatic event. For example, nightmares of a car accident or war experiences. And PTSD will be diagnosed if symptoms are present for over a month and if they really interfere with the well-being and daily functioning. But obviously people can also have subthreshold symptoms, so they may not necessarily fulfill the full diagnosis, but they may still have one or two symptoms. And especially this re-experiencing is quite common also in the absence of the full diagnosis. So how common is PTSD then? Well, this global survey that I just told you about also found out that the conditional risk for having PTSD upon one single trauma is on average 4%, but it really depends on the trauma type. So as you can imagine, interpersonal trauma such as sexual violence has a much higher risk for developing PTSD than, for example, experiencing a car accident. Moreover, what's also important to keep in mind is that with women are at increased conditional risk, even after accounting for trauma types, such as that women may be more often exposed to sexual violence. Even if you account for that, they're at increased risk. And this conditional risk combined with the prevalence of trauma results in about 12 lifetime episodes of PTSD per 100 adults in the studies. It's actually quite high. But luckily, it also shows from that actually following a traumatic event, the majority of exposed individuals, they actually will cope adequately or adapt after having some initial symptoms. And only this minority will develop long-lasting PTSD, and that can be either with immediate onset or a more delayed onset. So clearly there's a heterogeneity in the vulnerability or the susceptibility to develop PTSD. Carmen also touched upon that. I wanted to really emphasize that elucidation of these vulnerability factors and also the mechanisms associated with PTSD development, that's really highly prevalent for PTSD, because PTSD has the prerequisite of trauma exposure as its immediate trigger. It's also theoretically possible to provide preventive interventions really soon after traumatic events occur to prevent the long-term development of symptoms. So if we would elucidate these vulnerability factors, first of all, this could promote the early recognition of specific individuals at risk for PTSD following trauma, so that we can specifically target them with the early interventions. But also it could really lead to identification of novel targets for these preventive interventions. And this is the reason that over the past, I would say 2 decades or so, an increasing number of studies has really focusing on elucidating these vulnerability factors and also the etiological mechanisms of PTSD. And that's been done by performing assessments around the time of trauma and prospectively relating those to long-term clinical outcome. That sounds quite simple, but it's quite difficult actually to achieve, because only a minority of individuals eventually develops PTSD. You actually need quite high numbers of participants to in the end capture enough people with PTSD to compare them with. And also, I think you can also imagine yourself that's actually quite difficult to really capture people around the time of trauma, because in general, that's quite unpredictable. But there are some ways around it, and I will show you that in a bit. So the stress hormone cortisol and also its signaling via glucocorticoid receptors, that has been one of the, I would say, primary and most obvious candidates to actually focus on in relation to PTSD. PTSD was considered a stress disorder early on already, associated with prolonged, chronic, and extreme stress responsivity. It's even in the name, right? So therefore, it was hypothesized that cortisol levels would be chronically increased in PTSD patients, and also because of these re-experiencing symptoms that I just told you about with a clear memory component, and because cortisol affects glucocorticoid receptors in the hippocampal neurocircuitry, it was also really thought to be associated with these memory symptoms. But actually, the first findings in PTSD patients, they were quite unexpected. Because it was found that there were decreased instead of increased cortisol levels. And it was probably due to the fact that also increased glucocorticoid reception function was found. With higher numbers of receptors, increased negative feedback sensitivity of the HPA axis. And it was then thought that this decreased cortisol levels that were observed in patients might be a consequence of increased negative feedback, and that perhaps the acute stress response ending too soon. We also briefly already discussed that in the previous talk. So during my PhD, which is indeed 10 years ago, thank you, Sabine, I was involved in the first study to actually investigate worldwide whether this altered glucocorticoid receptor function could be a pre-existing vulnerability factor for PTSD. And it's already worked for quite some time ago, but I decided to discuss it here as my more recent work builds upon it. So here we actually found, I think, a smart way to come around the fact that usually trauma is pretty unpredictable by really focusing on a population that had a high risk of being trauma exposed in the near future. So this was done, this is the Prisma study, and was done in personnel deployed to Afghanistan, where they would have a high risk for combat and other trauma related and other traumatic events. This was worldwide the first study that really included a pre-trauma assessment of blood biomarkers. And I always like to emphasize, because I think the times have changed a lot, that this was all still hypothesis-based. So now we have the omics studies, which is much more data-driven, but here we really looked at specific candidates. So what we find, I would go briefly over it, is actually that we found in various ways, we assessed glucocorticoid receptor function in various ways. So we assessed the number, but we also expressed target, investigated mRNA expression of target genes, and we also investigated how sensitive glucocorticoid receptors in immune cells were for inhibition by dexamethasone, so synthetic cortisol. And each and every time, all these markers indicated that high signaling in glucocorticoid receptors already prior to deployment, so prior to the traumatic event exposure, predicted the onset of PTSD symptoms in response to traumatic events during the deployment, while basal levels of cortisol without any manipulation or challenge did not predict PTSD symptom development. So here you see the graph for the receptor number, but we also found the same for the receptor sensitivity. And then at the same time, there was some supporting evidence actually from other pre-trauma prospective studies and also high-risk populations that also found that PTSD development could be predicted by performing pre-trauma measures. And it's important to notice that basal or state cortisol levels were never predictive. So it was always either long-term cortisol levels or something measuring the reactivity during stress that was predictive, but basal levels were never predictive. And then also there were some early post-trauma prospective studies in which emergency department patients with injuries were investigated, which also found that if we do measures very early after trauma, we could predict PTSD development. So by measuring dexamethasone sensitivity suppression in the first days after trauma, but also then low circulating cortisol levels, if they're really measured really early after trauma, they were predictive of PTSD, but not anymore if we measured them later on. So integrating all this and also what Carmen just presented, it's really this PTSD vulnerability that seems to be related to pre-existing high glucocorticoid receptor function, which will lead probably to high glucocorticoid signaling during acute traumatic stress. And even if potentially peritraumatic cortisol output is not even increased, if it's the same between individuals, but one individual has more sensitive glucocorticoid receptor signaling, there will be a higher signal. But yet, actually, from these observational studies, we didn't actually know still whether cortisol reactivity actually during traumatic stress itself is also associated with PTSD development. So that's actually what I set out to do here together with Lotte Hilberding. She's the PhD student on this project. This is part of my VENI, indeed, as Sabine mentioned. So we wanted to investigate whether peritraumatic cortisol is associated with PTSD development. And for this, we set out to do an experimental paradigm. And we chose to do an experimental paradigm in healthy adults because this would allow us to really do repeated peritraumatic endocrine assessments, which is not possible during a real-life traumatic event. And then also still measure all the re-experiencing symptoms, which we're really interested in, or intrusive symptoms. Because obviously you can do a lot of manipulations in animals, but it's quite difficult to then really specifically investigate these re-experience symptoms, especially the content and also for how much distress it would lead to. So we used the trauma film paradigm. Basically, it's showing 30 minutes of very nasty graphic details of a rape. But it's been known to induce mild intrusive symptoms quite consistently, but I would stress mild and short lasting. So we can use it as a form of analog trauma to induce mild intrusive symptoms without inducing too much distress. And this trauma film paradigm is actually quite commonly used, but it's also known not to induce so many such robust HPA axis reactivity. And that's why we validated a novel protocol in which we added a psychosocial stressor just before looking at the trauma film. So here, this was the experiment. We had 63 participants. Half of them underwent a psychosocial stressor, the socially evaluated cold pressure task, while the other half didn't. And then they went all on to watch the trauma film. And then we measured their intrusive symptoms for a week with a smartphone application. So we could use ecological monetary assessment or real life monitoring. And then after 1 week, we got them back in the lab and they also filled out some additional questionnaires. So basically we find that indeed this adaptive paradigm lead to more robust HPA axis activation. So we saw higher cortisol levels just after the film and after the first recovery session. And then, although we did 6 assessments of cortisol, we had to divide them into separate phases for the next analyses because these were regression analyses. So we divided the cortisol in an acute reactivity phase. So that's, I can't see my cursor, that's too bad. But it's phases 2 and 3, and then 4, 5, and 6 were considered early recovery. And what we did find then was that higher cortisol stress reactivity with the AUC was associated with higher PTSD symptoms, but only in the cold water group, which had the more robust HPA axis activity overall, and not in the warm water or control group, and actually directions were opposite. And this was corrected for alpha amylase reactivity, and there was no effects of the cortisol recovery phase. Sabine, can you tell me how long I still have? Because for some reason my timer's not showing.
It's roughly 2 minutes, 2-3 minutes.
I don't hear. Sorry?
It's roughly 2-3 minutes.
Yeah, perfect. So then very quickly, so what we still wanted to do next was to see for these acute cortisol measures early after trauma, see whether we could actually take it to the next level and see whether we can go from, let's say, group level statistics to individual level prognosis. So can we use cortisol and other biomedical measures early after trauma to perform PTSD prognosis at the individual level? And we did this with machine learning techniques. So we included biomedical data collected as early as possible after trauma exposure upon admission to a trauma emergency department. Again, with this hypothesis-driven approach. So based on previous study, what did we identify there, and see if we can combine these data. And importantly, with eventual implementation in mind, we only selected features with really the potential for a routine collection of an emergency department admission. So this was done with machine learning study. And important here is to see that really these measures were taken in the hospital, and then individuals were followed up clinically over 12 months. So the overall model did very well at a high multi-class AUC, meaning that we were able to classify participants in their trajectories quite well. But let's zoom in on cortisol. So cortisol was actually the third most important features when we wanted to classify participants either into these trajectories, so immediate symptoms, delayed symptoms, recovering or resilient symptoms, but also looking at endpoint PTSD diagnosis. And interestingly, the directionality of the association, so lower cortisol as a risk factor, aligns with studies using more traditional linear statistics. But the classification rules, which you see here on the right, were nonlinear. So this is really saying that that these traditional statistics won't provide us with the whole picture. And also we saw nonlinear interactions with cortisol with features from other domains that were also relevant for the prediction. So for example, it's really interacted with whether participants really had the subjective experience of thinking they would die, whether they had experienced previous events, but also with, interestingly, the time of day the traumatic event occurred in, which is interesting because of cortisol's circadian rhythm. So let's wrap up. I think there's converging evidence from all these studies that individual variability in glucocorticoid reactivity and GR function is associated with PTSD development. We also have first indications that it can be a relevant contributor to individual risk prognosis, but it has really limitations in terms of the feasibility and the generalization. So this only holds for people who go to the emergency department early after a traumatic event, but that's obviously only a really small subset of individuals who develop traumatic events. And although it might be very nice to use such a model there, it's not so generalizable. It's important to notice, I would say, that all these studies, they make like snapshots of these inherently dynamic processes, right? So stress reactivity and recovery is a process in itself. And where you really measure it around the time of an acute traumatic event in terms of how long has it been since the traumatic event, but also the time of day, the exact measure you use, actually all of that will really influence the associations you'll find. And even if you do that repeatedly within an individual, the associations within persons may even change. And then also long-term, there's genetic influences. And again, all this GR function has been found to change upon previous trauma, chronic stress, aging, adversity, etc. And then importantly, I think it's been long neglected, but it's also really been shown that this is also really influenced by sex, but moreover also ovarian hormone status actually during the traumatic event may also be involved here. So clearly there's something going on, but it's really difficult also to compare findings between studies. Nevertheless, I think there's really converging evidence also if we combine the preclinical literature like Carmen just showed us, that this pathway may be really causally involved in PTSD pathophysiology, likely via hippocampus-mediated effects on memory. But it's really important. I think it's also nice to emphasize and stress here that really translational research will be crucial to elucidate these mechanisms. And nevertheless, it may be an interesting prevention target. Hydrocortisone is actually the most supported preventive intervention to date, but it might be also interesting to focus on intermediate cognitive mechanisms. Actually, Tetris as a visual spatial distractor influencing memory consolidation is promising, but perhaps also glucocorticoid receptor antagonist. It's important to stress that PTSD is a complex disorder. This will only be one of the many pathways potentially leading to PTSD, not the pathway. And it's really likely that we will need precision prevention. So we'll have to find out, and that will be the important challenge for the next year. Like what will work for whom should we target, and which intervention to provide. And with that, I would like to thank you for your attention. I can't hear you, Sabine.
Correct. Thank you, Mirjam, for your nice presentation. I see already two questions in the Q&A that kind of are similar, namely asking whether, cortisol from us by time of the day, was it the worst consequence in the morning or in the evening? And in fact, Livia asked a similar question, namely whether was it at awakening in PTSD or vulnerable individuals?
Yeah, so the time of day in the machine learning study, that was that actually individuals exposed at night were more at risk. And then I was super excited about that because I thought, yeah, that's super interesting because that's when cortisol is at its lowest. So it's really interesting in terms of the circadian rhythm. But then we also found out, which makes sense, that people exposed to traumatic events at night, they also really differ from those exposed during the day in, I would say, almost any psychosocial characteristic you can imagine. So they were more often male, they were younger, it was more often work-related or interpersonal violence. But cortisol seems to be involved there, and it was really denied. And then the second question, sorry, can you repeat that again?
So the second, oh, that's in the answered already. Well, there, let me see, that's in the, yeah, so it was regarding the cortisol levels at awakening in patients with PTSD or vulnerable individuals. So that is...
Oh yeah, so cortisol responses at awakening, we've been doing a longitudinal study in humanitarian aid workers where we really found that it's not necessarily this baseline pre-trauma cortisol awakening response, but how that changes in response to trauma that's associated with PTSD. So that seems to be more secondary, perhaps, to trauma and PTSD development, but I would say judge is still out there.
Okay, and then another question from Livia is, would you agree that patients admitted to the ER shortly after trauma might represent a specific subtype of patients? Because they, of course, they have a specific type of trauma.
Absolutely. So there's injury or suspected injury. So they're really, it's an important subgroup because injury and motorized vehicle accidents are super common. So even if it's still a small subgroup of the overall group, it's still relatively high numbers, but it's difficult really to generalize to the whole trauma population. Absolutely.
Okay. Then from Anke Toledo, is there a way to alter receptor number or sensitivity to prevent PTSD development? Or something else, that is, yeah. directly after the possible traumatic experience?
Yeah, so a long-term fix, I'm not so sure about, but what you could do is, for example, administer a glucocorticoid receptor antagonist early after a traumatic event to actually block the glucocorticoid receptor. That has not yet been done in human trials on prevention, but I would say the rodent literature is actually quite promising. So that might be one interesting way to go about if the side effects. For example, this is also used as an abortion medication in women, so we would need to be quite careful with that. But that might be one solution. The other way might actually be to administer cortisol instead of doing an antagonist to also sort of fill up the receptors.
You mentioned that the sensitivity of the receptor is altered, but is that the number of the receptors all are altered, or is there a natural polymorphism in the receptor that makes it more sensitive, or the affinity of the agonist in this case for the receptor?
Yeah, so it's several things. I actually deleted that slide. So there are functional SNPs, polymorphisms for the glucocorticoid receptor function, making it more sensitive, which indeed have been found to be associated both with PTSD and actually with glucocorticoid receptor number, also in our military study, interacting with previous trauma exposure. It only explains a very small percentage of the variance in the glucocorticoid receptor sensitivity, but it definitely contributes. It's also a repeated confirmed finding from several studies. And mentioning sensitivity, so this was measured in several ways. So glucocorticoid receptor number, binding affinity, and then functional consequences on leukocytes when once adding increasing doses of dexamethasone. So it's actually probably several ways, but I'm not sure whether they're all present in the same individuals or whether different individuals may differ in how exactly this sensitivity comes about, so to say.
Yeah, and cortisol might, of course, also interact with other receptors other than specific receptor for it, as we know it this far. Yeah.
Yeah, absolutely.
Yeah.
Yeah.
So then there is a question of Bogomil Peshev. Is there a connection between cortisol and sleep disturbance in PTSD, similar to psychotic depression?
That's a really good question. So there have actually not been so many studies really doing a fully circadian study on cortisol in PTSD, but those that have been done really seem to indicate that the most prominent disturbances in the circadian rhythm are at night for PTSD. And then it's a bit of a chicken egg question, I would say, because is it disturbed because there are so many sleep problems, or does it cause the sleep problems? I don't know about that. I don't think we're there yet, but it's really well conceivable, obviously, that it's associated with each other, but not necessarily causally, or we don't know yet, but it's very likely, I would say.
Okay. Brenda Pennings has the question, what about glucocorticoid reactivity as a predictor for PTSD treatment response? So yes, like EMDR, she mentioned.
Yeah, I think there have been some small studies setting out to investigate that, indeed also finding that it was a predictor, but to be really honest, I'm not sure about the directionality of these findings from the top of my mind. So whether increased sensitivity increased or decreased treatment efficacy. And I don't think, because I also see the question in the chat, that treatment has been found to change the reactivity yet, but it's not been so commonly studied as well.
Okay. And Kodrin had a question about memory functioning. So how is it impaired? Because there's lower hippocampal volume with higher stress reactivity. Leads to think that the memory has not yet been resolved, and it keeps bringing up the memory and its associated emotional valence to solve the traumatic experience.
Yeah, so what people think, what's now, so I would say the most common hypothesis in the field or the working model is that it's not necessarily that memories are over or under consolidated, but it's really the quality of the consolidated memory. So we now think that it's that traumatic memories are less well contextualized, so a bit more fragmented. So that people who develop PTSD, they also focus more on the peripheral details, the sensory details, without really consolidating the full narrative of the traumatic event. And if it's so fragmented, then it's quite easily to actually have this automatic spontaneous retrieval once you come upon a trigger of a traumatic event. So for example, if you were really focused on the red car and then you see a red car, it's quite easy for this red car later on to trigger your memory of the traumatic event, because you cannot really notice at the same time that it's actually not this traumatic event again and you're in a safe context. So we really think this contextualization, that's actually really at the heart of developing these intrusive symptoms particularly, and not be able to generalize the safety.
Sorry, that's also then generalization of the fear.
Yeah.
I would say. Yeah.
Yeah. So probably for really the content memory and the fear memory, that's probably the same processes, or at least that's one of the hypotheses now in the field, which is really mediated by the hippocampus. But this hippocampal volume, which is often thought to be a predisposing factor, these findings are a bit inconsistent, whether it's really a pre-existing trait. But it has been found in prospective studies that at least hippocampal functioning also predicts PTSD symptom development.
Okay.
Yeah.
So let me see. So there was another question of Sylvia. Did you see whether low cord reactivity predicted specifically PTSD sleep-related disturbances? And I think related to that, Huis says pre-trauma sleep problems increase the risk of PTSD.
Yeah, that's definitely true. Yeah, the last one. And for sleep problems per se, we have not investigated that so specifically for sleep problems only, but they do predict nightmares. But that's also intrusive memories. So, yeah.
Okay.
Yeah.
So let's move a little bit more to, you could say, the more general, perhaps overarching type of questions, in a way, in a more plenary discussion. And perhaps that's for both you and for Carmen a question. Like, because of course, you know, in a way, we're taking a look at vulnerability factors and predictors, which is of course very nice because you would like to have this more personalized medicine. You could, of course, include genetics into that as well. So apart from behavioral traits that you have, but then on the other hand, there's also the ethics component of it. So how do you value that? So what is your idea about that?
In the prediction of PTSD, you mean?
Yeah, because of course the thing is that if you classify people into, you know, like highly vulnerable and low vulnerable, is that a message that you would like to give them already? Yes or no?
I don't. So two things are important there. I think it's indeed more perhaps ethically. It makes more sense to really look at somebody's risk really after somebody has experienced something, and you do have the means to then monitor them very carefully and do provide them with interventions and help if they're in need of it. That's much more ethical, I would say, than determine one's vulnerability at a certain point in time and then saying, oh, by the way, if you do experiencing an event, you will be at risk. Because that will also do a lot in the anticipation, right? And have an effect on the stress response itself. So in that sense, I would always say, like, do it as a more reactive means. And then in terms of the genetic vulnerability, well, I think that the genetic studies, which all these combined cohorts, they're elucidating a lot of potential genetic factors involved, but the variance explained by these variants is still so very low that we cannot use it at this moment in time to really say something about individual risk. Perhaps we could, if we combine it with other factors, that would be interesting, I would say. But then I would always do it in a reactive way. If you really only have the means to really support people, once you tell them they're at risk.
Perhaps to add here, because our work is showing that the way people move, now it's in this virtual reality. We don't know if this can be generalized to normal life situations. But if this is telling so much, because movement now it's used for many different predictions also of maybe related to aging, but not only, as we see, it can tell a lot of things that eventually indeed maybe one single variable explains very little of the variance, but all together with these models can explain a lot. And then indeed, I think Sabine's point is very valid, because we're going to be able to know a lot. Like now it's happening with the face recognition. Nobody asks us if it's ethical or not. It's all over. I think perhaps this is something that we should be leading, to have more discussions of ethics, not only the ethics committees, but also scientists who are doing all this type of work and understand perhaps how far we can go. And otherwise, I think it will be late, because everything is coming very quickly. Genetics, it's everywhere as well. So yeah, I don't know. I think science is not bad on its own. It's the use that people can do eventually. So I think this is what we have to be careful.
Yeah,
right.
It really comes with responsibility.
In light of time, we'll have to close up the webinar, but not, of course, without prior thanking you guys both, Carmen and Mirjam, for this these great presentations, and to attend our webinar, to give your presentations. And let me share my screen for the final slide. And thanking everyone, also the audience, for joining us. If you want to watch this webinar again, it's recorded. You can find it at the TN2 website. And we currently invite you to join our next webinar on complex traits genetics on the May the 19th, the same time, again here on Zoom. So keep an eye also on our website, so TN2 website, and also on the socials, as on Twitter and LinkedIn. Thank you all, and see you next time.